So I was saying, welcome to everybody. This day will be dedicated nearly all to animal in science and research, but it will include also an additional lecture on basics of research integrity, which we will have this afternoon by Professor Taddeo from the University of Bologna. But my aim in this initial, not so long presentation will be to introduce a general issue of animals in research and in science. And I will do this by basically discussing quite briefly 3 points. First of all, I would like to summarize several of the issues which we already presented yesterday. That is the critical issues in preclinical research, and just summarizing several of the aspects which we discussed in previous lectures and focusing on the problems of animal research. And the second point is the critical one, that is, does animal research fulfill its role in biomedical research? And I will tell you why I decided to try to answer to this question. And finally, a Nuremberg Code for animal research. Of course, it's a provocative title, but maybe most of you know what is the Nuremberg Code, and I think that also based on the presentations that we listened to until now, maybe it's the case to speak about Nuremberg Code and also a Declaration of Helsinki, which, as you know, are the basic documents in clinical research, also for animal research. And so, first of all, the scientific research crisis. Yesterday we spoke very much about what we mean with this a few dramatic expression. I would like to very briefly summarize all the evidence that we saw yesterday, and there are a lot of reasons to think about the possibility that most of the biomedical research is in some way flawed. I mean, it's not, as maybe you understood from the several presentations, it's not only and merely a matter of core scientific integrity, but mainly possibly a matter of methodological weaknesses, weaknesses in the approach and the preparation and the planning of studies, and then in their analysis and in their presentation. So maybe the most representative paper in this regard, in my opinion, it is still the provocative paper by the John Ioannidis, this worldwide renowned American epidemiologist, who published nearly 10 years ago this still very, very highly cited paper, which he entitled Why Most Published Research Findings Are False. And as we saw yesterday, he wrote that a research finding is less likely to be true when the studies conducted in the field are smaller. I mean smaller, first of all, in terms of sample size, when effect sizes are smaller, when there is a greater number and lesser preselection of tested relationships. That is, now it's possible to say, when we are going to p-hacking, as we saw yesterday, when there is greater flexibility in design, definition, outcomes, and analytical modes, and when there is a greater financial and other instances prejudice. And we saw yesterday also some examples of disclosures of conflict of interest, which actually occurs in a very small percentage of published papers. And when more teams are involved in scientific fields in chase of statistical significance, which means also, and we already saw several examples in this regard, that competition in science is not always the best approach, and so that's it. We already saw yesterday also this figure which was used by Professor McLeod to exemplify and to provide a scheme of the several aspects which may produce, as he told, waste in biomedical research. starting from the questions which we decide to address in our research, to the designs and methods which we use in both preclinical and clinical studies, to the publication or the failure to publish the results, maybe because they are just negative results, maybe because they do not completely fulfill our expectations, and also the possible bias in the reports of obtained results. Yesterday, Professor McLeod, in relation to animal research in particular, to animal research in the neurological sciences, provided a lot of examples and also several possible approaches to try to fix in some way the situation. But in any case, the point is that even if these flaws are in some way general, they affect biomedical research as a whole, both on the clinical and on the preclinical side. Now I would like to provide you some elements just to show that it's possible in the preclinical field, and maybe in particular in animal research, that most of the critical points arise. These are exactly all the papers that we commented yesterday, just showing that there is some reproducibility crisis, as we called it, in which particular affect, or maybe just because all these researchers focused on results from animal research. So one would say, why animal research? Because in particular in drug development, but also in basic research, just addressing the pathophysiology of biological systems, usually animal models are considered in some way, let's say, the golden standard, or at least the place in which to check the hypothesis which we can build based upon in vitro models. Of course, it's also a matter of complexity. There are some biological phenomena which cannot be studied unless we use complex models, and animal models are still now considered an unavoidable step in translating basic research into clinical evidence. And so this is why all people focus on animal models, and maybe this is also why most of the stuff related to the problems in affordability, quality, and reproducibility of experimental results now arise by a careful examination of the results in animal studies. And possibly all of us who have a few knowledge of, for instance, of the process of drug development are used to think in terms of the animal models as the last and the most important step in the translation of basic evidence to the clinical setting. But we will see that there is more than some evidence that this is not actually the case. And this is one of the aspects that in some way affect presently the use of animals in science. And we also saw as an example this paper, which I also mentioned yesterday. I think that it's very interesting and provocative, since, as I already told you yesterday, it provides some evidence that preclinical research in general, but this paper deals exactly with the use of animal models for a specific purpose, that is the assessment of efficacy of statins in atherosclerosis. I mean, preclinical research is in some way peculiar in that it is affected by factors which can be a few different from the factors which are now well known to affect clinical research. We saw, for instance, that this paper identified as a bias, as one of the main biases possibly affecting the reporting, but also the effect size itself, which is found in experimental studies, the origin, the context in which the study was performed, but this is the opposite of what usually happens in clinical research, because yesterday we was discussing how clinical research is usually in some way biased, for instance, by the financial interest of sponsors and promoters of the research, in particular when the studies are concerned with the development and registration of novel medicines, and so when money comes from the pharmaceutical industry. And there are a lot of papers showing how in this case there is a high probability that results will be more favorable to the experimental drugs the more as it is important the financial interest of the sponsor. But in this case, as highlighted by these red circles, it seems the opposite in that in this meta-analysis of animal studies, the effect size is significantly wider, not in industry-sponsored studies, but in academic-based studies. And yesterday we mentioned very often the paradigm of publish or perish, which is actually very a big pressure and a big push on, in particular, on academic researchers. And possibly this is an evidence, an indirect evidence of the possible effects and distortions and biases which this paradigm is imposing in academic research, which is which should be paradoxically, and we used to think about academic research most of all in terms of more autonomy, more freedom, more independence, which overall should result in more objectivity. But maybe this is not always the case, and these are factors and phenomena which we need to take into account. So, the main questions of this introductory presentation. I mean, does animal research fulfill its role in biomedical research? That is actually animal research, the use of animals in science, the core point, the main instrument which allows us to translate findings in basic preclinical research to the clinics and maybe to applications in therapeutics. And this question in some way resembles the title of some provocative papers which were published over the last about 10 years in several major journals, starting from the British Medical Journal, which, as maybe now you can understand, is one of the journals which are more and more maybe most sensitive to the issues of quality, of ethics, of objectivity, of transparency in science, both in preclinical and clinical science. And I recommend you very much to consider this journal among the journals which should be routinely read by not only clinical but also basic researchers. And this is a paper which was published now exactly 10 years ago, in 2004. It was authored by Pandora Pound, who is a medical sociologist, together with Jan Roberts and some collaborators. And the title was very provocative. The point was, where is the evidence that animal research benefits humans? And what they did in this paper, I warmly recommend you to consider this paper, because for me it was very useful when I found it, when I read it. They just go through the available literature, looking for systematic reviews and meta-analysis of animal studies. Yesterday, Professor McLeod explained us the definition of systematic reviews and meta-analysis for comparison to narrative reviews. So systematic studies of papers available in literature concerning what it was done in selected critical areas of biomedical research with a high relevance for the clinics. And they found and examined in this paper these 6 areas of research where they found actually systematic reviews of animal research. You see, calcium channel blockers for stroke, fluid resuscitation for bleeding, thrombolysis for stroke, that also Malcolm McLeod told us about yesterday afternoon. And these are their conclusion. So first of all, the value of animal research into potential human treatments needs urgent, rigorous evaluation. Systematic reviews can provide important insights into the validity of animal research. This was written in 2004, and yesterday you saw several examples of what has been done in the meanwhile. I mean, for instance, the presentation of these international networks dedicated to the systematic building of meta-analysis in animal research. But at that time I wrote, 10 years ago, the few existing reviews have highlighted deficiencies such as animal and clinical trials be conducted simultaneously. It's exactly what I told you. For instance, the calcium channel blockers for stroke. It's quite disappointing to note that if we go and see, we see that the clinical research was actually not based on evidence from animal studies. So one would wonder, but why doing animal studies in this regard if we are already at the clinical stage? The poor methodological quality was discussed thoroughly and deeply discussed yesterday afternoon. And so systematic review should become routine to ensure the best use of existing animal data, but also they should become routine also to highlight the flaws and weaknesses of animal studies and to improve their quality. And which were the main methodological problems? In some way, most of them we already saw yesterday in the field of neurological sciences, but it is a general issue in many fields. I dare to say in nearly all the fields of preclinical research, that is, different animal species strains. Of course, the ones of you who are familiar with the use of animals in research know that only animals which are selected exactly for scientific research and which are usually inbred, so they are highly genetically homogeneous animals, but in any case a lot of different strains exist, so which is the relevance of and I would like to say that maybe the same strain, but the same strain, an issue which is relatively still under-investigated in this field. The same strain provided by different producers. I will tell you later that this is an issue which has been already characterized as critical in the use of cell lines in vitro research. But still very few people just investigated the possible differences in the same strain, but provided by different producers. And the different models. Still, again, yesterday we heard about different protocols, different drug dosing schedules and regimens, the variability in the ways animals are selected for studies, nearly never by randomization, nearly never with a clear controlled design, nearly never with blinded assessment. In the clinical research, we are absolutely familiar with the need to use blind assessment of patients, either single-blinded, possibly double-blinded, so that's also the physician assessing the patient is not affected by any prejudice or bias. But in animal research, this is quite the completely unknown practice. We can just look at animals and we rely. But again, yesterday you remember maybe the example of some behavioral tests in mice and how the outcome could be affected by the expectancies of the observer, of the evaluator. And again, the statistics, friend or foe, agony or ecstasy, inadequate power, no calculation of power at all, simplistic statistical analysis, just looking for some significant p-value, failure to follow the intention to treat principles. The intention to treat principles, that is, for instance, failure to tell about the actual number of animals which was originally included in the groups, just providing data only about the animals who completed the study. In clinical research, all of us just would say that a clinical study which provides data only about patients who completed the study is seriously flawed and absolutely not clinically significant at all. But in animal study, who cares? Okay, I lost some animals, so what to do? I will put maybe another cage with additional animals and we'll see what it will happen. And in the end, it will be the same amount of animals. The variety, the outcome measure, the length of follow-up. So a lot of aspects clearly identified as critical, but still now need to be properly addressed. And so in the meanwhile, a lot of papers. In this case, I present mainly papers from not randomly chosen, of course, because the groups with whom Professor McLeod collaborates are among the main ones which are providing evidence for the need of improvement in methodological approaches. And his conclusion yesterday, which is based on a great amount of experimental research, of work on available evidence, is that systematic reviews of animal studies reveal evidence of selective analysis, outcome reporting bias, as well as publication bias, leading to overstatement of the validity of the entire body of research, and maybe, as you saw yesterday, overstatement of efficacy and understatement of possible risks from animal studies. So I also would like to say, from my personal point of view, this is mainly a much more useful critical approach to animal research in science, and much more useful, important and productive, for instance, than the approach of animal protection, let's say so-called protection organizations, which usually just fight on the basis of some ideological and emotional reasons. These are just a lot of scientific reasons to deeply rethink about the use of animals in the research, and which could provide, first of all, the possibility to strongly reduce the animals used in science. Yesterday we saw that in the field of neurological sciences, nearly about 50% of the animals which are used, is possible to say that they are just wasted, just because they provide no useful information. So 50%, it's a huge amount of animals. And anyone seriously concerned with protection of animals, with animal care, should think first of all about this kind of approach, which would also improve, of course, the outcome of experimental research. And this is another paper which was published 10 years later, again from the same Pandora Pound which I mentioned yesterday, this medical sociologist. By the way, we also invited the Professor Pound to give a lecture in this course, and it was not possible for her in this period, and we strongly hope that it will be possible in a future edition of this course, just because of the contribution that she's giving to this very, unusual and provocative point of view on the topic of animal research. And in this paper, she is asking, is animal research sufficiently evidence-based to be a cornerstone of biomedical research? Of course, the answer is not a full yes, but maybe is most of all, yet in some way more no than yes, due to a lot of critical aspects. In this paper, a lot of initiatives which arose in the last few years is mentioned and described. For instance, the development of some guidelines for reporting of animal studies, this ARRIVE, Animal Research Reporting in Vivo Experiments. This is the first attempt to develop a guideline we we used to know for clinical studies, and which we will talk about in the next few days in several lectures dealing with clinical research. Up to a few years ago, there was no such guidelines in clinical studies, and now this arrive is the first example in this regard. They were developed in 2010. And also in the Netherlands, in particular, this organization, the Systematic Review Center for Laboratory Animal Experimentation, the Circle, developed this gold standard publication checklist. It's something similar to this ARRIVE guidelines. And also in the UK, this organization, which is the main not-for-profit organization in the UK for the replacement of animals in medical experiments, also provides free online courses and workshops in experimental design of animal studies. So a lot of initiatives which are focusing on the possibility to improve the quality, the accountability, the transparency, and finally, of course, the relevance and the potential of animal studies for biomedical research. Because in any case, the conclusions at present are still the same. The conduct, reporting, and synthesis of much animal research continues to be inadequate. This current situation, of course, is unethical, since animals and humans participate in research that cannot produce reliable results. So one wonders, why doing this kind of research? The last question, the Nuremberg Code for animal research. Of course, it's very, very provocative, this I understand, because as you know what it was, the Nuremberg Code. But why the Nuremberg Code? Maybe you remember yesterday, but also Professor McLeod mentioned about several flaws in published reports of animal studies, showing what happens over the years. For instance, what happens over the years about I mean, in papers dealing with animal studies concerning proper description of the experimental design, the randomization, blinded procedures, etc. And he showed that only a few percentage of papers reported these details, but situation didn't change over time. I mean, even if a lot of people now step by step are aware of these weaknesses, nearly nothing changed in the last decades. The only aspect that maybe changed or apparently changed in some way is reporting of conflict of interest. Maybe you remember this slide. I think this is exactly representative of what I would like to tell you in this last point. And yesterday it was underlined, emphasized how this disclosure of conflict of interest is possibly a few improved over the years, but just because journals require that when you submit a manuscript, you will disclose your conflict of interest. And usually when you submit a paper, if you not disclose conflict of interest, your manuscript will be not even evaluated, and just you will receive it back with the kind request to include this very short sentence stating if you have any conflict of interest. Conflict of interest in biomedical research, both clinical and clinical, is critical. We will have a specific lecture tomorrow by Alice Fabri in this regard. But now I just mentioned this just as an example that if anything improved in this regard, maybe it's possible to say that improved just because someone who was in the position to do it decided to introduce a new rule, a new requirement, a requirement which was systematically required. So maybe we should do something similar also in animal research. Another point of view, again, this paper published in Nature with the viewpoint of the NIH on this reproducibility crisis in biomedical research, and what we should do. We mentioned yesterday this paper, and again today we mentioned this paper. What they say, it's another quote from this position paper, and they say, preclinical research, especially work that uses animal models, seems to be the area that is currently most susceptible to reproducibility issues due to different animal strains, different laboratory environments, subtle changes in protocols, all the issues which we examined yesterday and we summarized today. Of course, it's not a matter of finding who is guilty of anything. Some irreproducible reports are probably just the result of coincidental findings that happen to reach statistical significance, coupled with publication bias and coupled also with some different methodological bias, which are still honestly, but of course not acceptably performed by researchers. Another pitfall is over-interpretation of results and discovery studies. And in any case, there remains a troubled frequency of published reports that claim significant results but fail to be reproducible. But it's interesting that also in this position paper, they immediately do a comparison with what happens in clinical research. Reproducibility is potentially a problem in all scientific disciplines, of course, in all experimental disciplines. However, human clinical trials seems to be less at risk because they are already governed by various regulations that stipulate rigorous design and independent oversight, including the need for randomization, the need for blinding, the need for power estimates, the need now, over the last decade, for pre-registration of outcome measured in standardized public databases, clinicaltrials.gov, but also in Europe, a closed database which is run by the European Medicines Agency. You can do no clinical trial, no clinical studies, unless if this is a drug registrative study, you register the protocol in advance in these databases. That is, everyone can know that you are going to do this study, and then you should be bound to tell what happens. No matter publication. I mean, publication can be a second step. This is just preliminary, let's say, I would like to say informal, but this is formal and official place in which to publish, or at least in which you can make accessible the results of your research, independent of publication. It's also a way, in some way, to overcome the publication bias with which we deal. And the oversight by institutional review boards, data safety monitoring boards, etc. And a lot of procedures, which are now standard procedures as imposed by a lot of guidelines, such as, for the one of you who is familiar with clinical experimentation, the good clinical practices, or as I told you, the Declaration of Helsinki, and a lot of national laws which put into action the international guidelines which are developed to render accountable, rigorous, verifiable, and possibly, of course, reproducible the results of clinical research. So why a Nuremberg Code? Just because, for animal research, I mean, just because the Nuremberg Code was, maybe it's possible to say that it was the initial document from which all the subsequent regulatory statements on clinical research just began with. And the Nuremberg Code, as maybe you know, is a set of research ethic principles for human experimentation set as a result of the subsequent Nuremberg trials at the end of the Second World War, in particular the doctors' trials against the doctors who were in concentration camps, in Nazi concentration camps. And this was the initial document which included the main principles, which still now, of course, after a lot of development, a lot of elaboration, a lot of more detailed discussion and specification, are just the basic principles upon which all the also methodological and scientific approach to the clinical research is based. Of course, a lot of principles, but in particular, as I emphasize here, properly formulated scientific experimentation that is clinical studies need to be founded on a strong scientific background, on careful assessment of risks and benefits, a reasonable likelihood to benefit, okay, to the population studies, must be conducted by trained investigators under the responsibility of a physician, using approved protocols, and these protocols need to contain all the information, including assessment of statistical power, preliminary definition of outcomes, detailed description of the procedures of randomization, blinded assessment, everything told in advance, sample size, and all these issues which now we used to think are just fundamental to ensure the quality of the results of a clinical study. Ethical publication extends to publication of the results and consideration of any potential conflict of interest. And this is just arised from the Nuremberg Code that later on, Nuremberg Code was issued in 1947. Later on, about less than 15 years later, at the beginning of the 60s, the Declaration of Helsinki in its first version was issued. We are now at the 8th version, and this is still the main reference document for clinical experimentation, and which gave rise to a lot of practical guidelines for human experimentation. So possibly we should start to think possibly about a similar set of rules and guidelines also for animal experimentation, and possibly we are exactly going step by step and very slowly, but into this direction. These are just some papers which were published very recently, nearly all this year, just to say that in any case, worldwide, attention and concern of the scientific community is increasing worldwide in this regard. You see this paper in PLOS ONE, the need for the randomization in animal trials, or this perspective, all preclinical trials should be registered in advance in an online registry. And in Europe, it's maybe possible to say that it's the UK, the nation in which more attention is being devoted over all these years, and of course the BMJ is published in the UK and is mainly concerned with what happened in UK for several aspects. And in UK, it's maybe the place in which the discussion has evolved more over the last few years, and this is a news which was reported in the BNJ just very recently. You see a group of 72 organizations, including a lot of universities, all organizations that carry out, fund, support animal research, have signed a concordat committing themselves to being more open and honest about how and why they use animals in their research. And in particular, universities, their companies, societies, etc., have signed up that they will be expected to encourage researchers to follow the good practice outlined in these international guidelines on how to publish animal research. So step by step, we are going into this direction. There were several meetings, several congresses. It's possible to say, for instance, that researchers working with animals, of course, are still a few not so happy with the perspective to be burdened with additional rules that now they do not need to fulfill. But in some way, I think that at least in animal research, we should go towards this direction, or in any case, we should go. It's a matter of fact that the situation is evolving exactly in this direction. So this was the meaning of the Nuremberg Code for animal research. And I would like to end this very introductory presentation just saying that, of course, we are focusing on animal research. We decided for this first edition of this course to focus as regard preclinical research, mainly of animal use in science, just for the reasons that we widely discussed also today. But there is also a lot of concern for preclinical in vitro research for several aspects, which in my opinion are very well summarized in this paper, which which was published last year, Food for Thought, Look Back in Anger, What Clinical Studies Tell Us About Preclinical Work, where all the concerns about animal studies are again summarized, but where also a lot of concerns for in vitro models are discussed. For instance, as I told you, the problem of the homogeneity of the cell lines which are used as in vitro models for cells and tissues, in particular, but not exclusively in cancer research. And there are a lot of studies, but also very old studies that show and warn researchers about the possibility that what they are using bona fide as a kind of cell actually result to be a completely different kind of cell, maybe just because of contamination of the cell line which they have by a different cell line, a contamination that may happen not in our lab but in the producer's facilities, or also because of spontaneous mutations of the cell line. I mean spontaneous mutations which may happen at any stage of this immortalized cell line's lifespan. So what we call HeLa, what we call MCF7, what we call CHO, etc., it may result to be completely different cells. And there are a lot of papers in this regard, for instance, showing that a lot of in vitro studies on many kinds of cancers, for instance, prostate cancer, breast cancer, etc., are basically completely flawed just because they used completely different kinds of cell lines. So just to say that I think that in the future we will need to devote some attention also in courses like this for the training of young researchers, also on the critical issues which may affect in vitro models. So that's it. And just to conclude with a few smiles, but just a few smiles. This is a citation from a website that is PhD Comic. I don't know if any one of you knows about this website. It's a website which publishes on nearly a weekly basis comics about the life and activity of PhD students working in some university in the world. And this was a citation just dealing with how to write a honest method section. And you see in this method section of the paper they say animals were treated in accordance with our funding agency's animal care and use guidelines and in compliance with IRB protocol. So we are doing very well and very careful with animals. But the students, however, were treated terribly and paid very little. In fact, we worked them pretty hard. This was done in accordance with the fact that they kept showing up to work, and so why not? But why not? Yesterday we, most of you, Natasha, for instance, underlined in discussion, so the very, very critical issues of young researchers, and this course is dedicated to the training of young researchers. All of you, I think, if you are here, it's just because you think of yourself in terms of young researchers, and maybe you expect, I think most of you or some of you, that it will be possible in some way to have a career in research. And this, of course, is a big problem. Not only in terms of possibility of personal exploitation of your work in the labs, but first of all in terms of the general organization of research, not only at the national level, but also worldwide, and the perspectives that can expect young people who would like to engage themselves in research. And in this regard, after the discussion of yesterday's discussion, yesterday evening, I was a few struck by this paper, which was very, very recently, just a few days ago, 7 of November 2014. It was published in The Guardian. It's one of the main newspapers in the UK, and you can find it in The Guardian online. And it's a very interesting paper, which just criticizes very strongly the organization of Horizon 2020, that it's presently the biggest program supporting research, not only in Europe, but worldwide. It has about 68 billion euros available for research in the next few years, and a lot of money is put on the support of young researchers. But according to this article, which I would like to say I support very much, its criticism and conclusion, the basic rules of these programs are actually too much focused on what they call excellence and what they believe to be meritocracy, but just giving very few prizes to very few people. So these are the proposals. For instance, they say, what about reducing the size of ERC grants for young researchers and increasing the number of grantees? Maybe young researchers do not need a lot of money and to build immediately a research group with also a lot of collaborators, but need first of all to continue to have support in their career and to strengthen their publication record. What about introducing ERC basic fellowship with just a long-term salary for applicants who would like to do research on their own projects? And also it's very interesting, there is also a criticism to the emphasis on mobility. Of course, mobility is very important in research, but now an increasing number of people begin to think that mobility is important, but maybe possibly more for senior tenured researchers rather than for young researchers. And there are studies which increasingly show how this forced mobility to obtain this money from the European Council, if it is forced, that is, it forces young researchers to move from one country to another. If this is not functional to the career of the researcher, it may also result in some damages. So this was an article which in some way claims to for a rethinking of the programs which should support the career of young researchers in the near future. So, I think that's it. Thank you very much. Questions, comments? I think maybe no, or something. Okay, I think this was just an introductory presentation, so I think we can stop here.